we are left with only one topic, apomixis as well as polyembryony, that we will complete by today. So apomixis means development of vegetative propagules without meiosis and syngamy. So apomixis is basically development of vegetative, or you can also write development of reproductive propagules without meiosis and syngamy. That is, no meiosis, no syngamy, no fertilization, even though embryo is formed. If embryo is formed in such case, then it will increase in the number of the embryos, and that condition is what we basically call it as polyembryony. So apomictic embryo, the embryo that is developed using this process. Which process? Apomixis. What is apomixis? Development of the reproductive or propagules, that is basically nothing but the embryos without meiosis and syngamy. No fertilization, no meiosis. Right? So apomictic embryos, if develops, it increases the number of embryos in the seed, and such condition in which more than one embryo is present is called as polyembryonic, and the seeds are called as polyembryonic or apomictic seeds. Right? So there will be one fertilized embryo and one that would be developed by apomictic, that is apomixis embryo or apomictic. Right? So polyembryony is basically occurrence of more than one embryo in the seed. Example includes, you can take the citrus fruits, Apuntia you can take mango, you can take onion, groundnut as the best example of this polyembryony. Right? Now apomixis is basically, it is mimicking the sexual reproduction, but basically it is not actually a reproduction of sexual life. So it mimics sexual reproduction. Why? Because embryo is formed without meiosis and syngamy. Let us see the types also. So apomixis is basically divided into two, that is agamospermy and vegetative propagation. Vegetative propagation is because no meiosis, no syngamy, that we have already discussed in chapter number 1. Right? So we will not discuss now. And this is agamospermy. Agamospermy is no meiosis, no fertilization. Still embryos are developed. Right? Now let us discuss about the types of agamospermy. This agamospermy is again subdivided into 4, and these 4 are very important for your competitive exams. So adventative embryony or adventative polyembryony, whatever you want to say, you can say. So adventative embryony, right? Then the second is apospory. Third one is diplospory, and the fourth one is pure or parthenogenesis. So vegetative propagation of the apomixis, agamospermy, no meiosis, no fertilization. 4 types: adventative embryony or adventative polyembryony, the same condition, more than one embryo; apospory, diplospory, and parthenogenesis or parthenocarpic foods. So now let us discuss one by one. First is adventative embryony. What happens in the adventative embryony? Adventative, that is addition, other than the normal cell embryos So formation of embryo directly from the diploid sporophytic tissues, basically either nucleus or integument. So it is development or the formation of embryo. You can write development or formation of embryo directly from nucellus or integument of ovule. Right? Here again, meiosis is not taking place, fertilization is not taking place. Directly from the nucellus or integument of the ovule, embryo is developed, and basically it is from the diploid sporophytic tissue, or you can say diploid sporophytic cells, which are either nucellus or integument. Example includes citrus, opuntia, anyone you can learn. Right? Second is apospory. Apospory means it is formation of the embryo sac, not embryo. Here embryo was formed. In apospory, embryo sac is formed directly from the diploid sporophytic cells, either nucellus or integument. So apospory. What happens in the apospory? Development or formation of embryo sac from again diploid sporophytic cells, that is either nucellus or you can also write integuments. Example includes Parthenium, that is basically carrot grasses or the normal grass. Parthenium. So this is apospory and adventative embryo. Once again, let us discuss. This Apomixis types are of two: vegetative propagation, that is vegetative reproduction, that we've already seen. No meiosis, no fertilization, because only one parent is involved. So agamospermy, no meiosis, no fertilization takes place. Types: adventative embryony, apospory, diplospory, parthenogenesis. In case of vegetative propagation, we know only one parent is involved. That is, in case of plant, the vegetative structures are involved. So adventative embryony, apospory, diplospory, and parthenogenesis. Adventative embryony and apospory, the difference is only one. In case of adventative embryony, the embryo is formed from the sporophytic, diploid sporophytic cells, that is either the cells of the nucellus or integument. Here embryo sac, which is again a diploid structure, is formed. Within the embryo sac, embryo will also be developed from the embryo sac, right? So this is the only difference between adventative embryony as well as apospory. Now the third is diplospory. What happens in the diplospory? Here MMC, megaspore mother cell, develops into embryo sac. MMC develops into embryo sac. What is MMC? It is megaspore mother cell, because we are discussing regarding the embryo and the ovule part. Right? Embryo formed from unfertilized egg. Example you can take dandelion, right? Example is dandelion. And fourth, you all are aware of, that is parthenogenesis, formation of embryo from unfertilized egg, right? Or seed. So partheno, that is formation of embryo from unfertilized egg. That is what we basically call as parthenogenesis. You can take any example. We have example of parthenogenesis, banana. You can take example of watermelon. Now in such fruits, the seed will not be there because they are seedless fruits. So this was all about apomixis as well as polyembryony. Four are very important: adventative embryony, apospory, diplospory, parthenogenesis, and one or two examples of each class. let us now discuss about the significance or the importance of apomixis. So why apomixis is important? So in order to preserve or maintain the desired characters preserved for the generation, this apomixis phenomena is important because here no meiosis, no fertilization is taking place. Say for example, the best example that I can give you is the pollinators. Different pollinating agents are used, right? Pollinators, and that is a pollinating agent that are coming from different plants, right? Different plants have different pollen and different pollinating agent is required. So all the time pollinating agent may be different, pollen type will also be different. So the continuity, that is the variety, the desired species will not be maintained. But in case of apomixis, that desired character will be maintained. So desired characters get preserved for generation, because only through apomixis only the embryo will be developed, right? And here no meiosis, no fertilization takes place, so that is an advantage. So it has a great significance in agriculture, for production of parthenogenic seeds. Production of parthenogenic seeds. You see this banana and now the watermelon are also coming parthenogenic. Tomatoes also seedless. Then no need for pollination. For fruit and seed development. See, because pollination is not at all required. So different pollen grain and different pollinating agent, right, are involved in the normal pollination. That is what we basically call it as a cross pollination. Here pollination is not taking place, so there is no point of different pollen grain coming from the different plant and different pollinating agent that transfer the pollen grain, because meiosis is not taking place, fertilization is not taking place. So what is the use of pollen grain? So this was all about apomixis and polyembryony.